Growth is an increase in size of a tissue or organism due to increase in cell size. This is called hypertrophy. Increase in cell number. This is called hyperplasia. Or increase in extracellular matrix. Today, we will cover 5 topics. First, we will look at growth hormone. Growth hormone is also known as somatotropin. It is a major hormone regulating growth in the body. It is a peptide hormone with 191 amino acid residues. It is synthesized by somatotropic cells of the anterior pituitary gland. Growth hormone receptor is an enzyme-linked receptor. It is most similar to cytokine receptor. The receptor does not have intrinsic kinase activity. It is associated with a separate intracellular tyrosine kinase, Janus kinase 2, or JAK2. JAK2 is encoded by a different gene. Binding of the growth hormone to the receptor results in dimerization of the receptor. JAK2 then phosphorylates receptor, which then recruits START. JAK2 then phosphorylates START molecules, which dimerize and move to the nucleus. START stands for signal transducers and activators of transcriptions. START is a transcription factor, a member of the START family. About 50% of growth hormone is bound to growth hormone binding protein. Bound growth hormone is not biologically available. Therefore, it decreases biological activity of growth hormone. But growth hormone binding protein increases the half-life of growth hormone. Growth hormone binding protein is identical to the ligand binding domain of growth hormone receptor. It is derived from alternative splicing of growth hormone receptor RNA. Here is a primary transcript of growth hormone receptor gene. When the transmembrane domain is retained, the RNA will code for growth hormone receptor. Through alternative splicing, when transmembrane domain is lost, the resulting RNA will code for growth hormone binding protein. Growth hormone has both catabolic and anabolic functions. First, growth hormone has direct catabolic effects. It decreases uptake of glucose into adipose tissue and muscle. As a result, it raises blood glucose level. It also mobilizes fats by increasing the activity of hormone-sensitive lipase. As a result, it raises the blood-free fatty acid level. Second, growth hormone has a direct anabolic effects. It increases uptake of amino acid into cells. Third, growth hormone has an indirect anabolic effect. It increases synthesis of insulin-like growth factors, or IGFs. Here is a comparison between anabolic hormones and stress hormones. Anabolic hormones tend to promote protein synthesis. Anabolic hormones include thyroid hormones, growth hormones, or IGF-1, sex steroid, especially androgens, and insulin. Stress hormones act to mobilize energy stores. The stress hormones include growth hormone, glucagon, cortisol, and epinephrine. This slide shows you the function of growth hormone in control of growth. Growth hormone is secreted by anterior pituitary gland In plasma, growth hormone is bound to growth hormone binding protein. After it arrives in the liver, growth hormone binds growth hormone receptor and activates the growth hormone receptor pathway, which increases synthesis of three target genes. One is IGF-1, second gene is ALS, third gene is IGF binding protein 3. These three proteins form a complex and circulate in the blood. IGF-1 is released from the complex by IGF binding protein 3 proteases. When IGF arrives in the bone, it binds IGF receptor, which in turn promotes bone growth. Therefore, growth hormone controls skeletal growth indirectly by regulating synthesis of IGF-1.
Now, let's move to part 2. We will look at insulin-like growth factors.
Growth hormone controls skeletal and muscular growth through IGFs. Specifically, growth hormone acts on the liver and promotes synthesis of IGFs, and IGFs control growth of bone and muscle. In particular, IGF-1 is required for growth before and after birth. IGF receptors are very similar to insulin receptor. Therefore, they are receptor tyrosine kinase. Insulin can also bind IGF receptor, although with a lower affinity than IGFs. Several hormones work together to control growth at different stages. At embryonic stage, intrauterine growth requires growth hormone, IGF-2, IGF-1, and insulin. Infants of diabetic mothers are typically large because they have been exposed to high insulin levels. Postnatal growth requires thyroid hormones and growth hormone. Hypothyroidism following delivery causes irreversible abnormalities in nervous system maturation. Acquired hypothyroidism later in childhood will slow growth and reduce bone advancement. Prepubertal growth hormone deficiency leads to dwarfism. Hypersecretion of growth hormone pre-puberty results in gigantism. At puberty, there are reproductive changes. Hypothalamic pulse generator increase activity just before physical changes at puberty. First noticed sign in a female is breast development. First noted sign in a male is enlargement of testes. At puberty, there are also growth changes. Androgen promote growth in the following anabolic sequence. When thyroid hormone is normal, a rise in androgen increases growth hormone secretion, which in turn increases IGF-1 secretion. IGF-1 promotes division of cartilage synthesizing cells. After puberty, growth hormone is only needed to maintain lean body mass. On the other hand, a high level of growth hormone can cause acromegaly.
Now, let's move to part 3. We will look at regulation of growth hormone secretion.
One important hormone that controls growth hormone synthesis is growth hormone releasing hormone. It is a peptide hormone produced by hypothalamus. Growth hormone releasing hormone binds growth hormone releasing hormone receptor on pituitary gland. Like most G protein coupled receptors we have seen so far, growth hormone releasing hormone receptor also use G-alpha S cyclic KMP pathway to activate transcription of growth hormone gene. When growth hormone releasing hormone binds as a receptor on somatotroph, it activates G-alpha S adenylate cyclase and protein kinase A pathway. A target of this pathway is PIT1. It stands for pituitary-specific positive transcription factor 1. It is a transcription factor. Activated PIT1 increases transcription of growth hormone gene. Somatostatin binds somatostatin receptor on the same cell. It is also a G protein coupled receptor. Binding of somatostatin activates G-alpha I. G-alpha I inhibits growth hormone releasing hormone receptor cyclic KMP pathway, whereby somatostatin inhibits PIT1 activity. Somatostatin is secreted in the digestive system, such as delta cells in the pyloric antrum, duodenum, and the pancreatic islets. It is also produced by the ventral medial nucleus of hypothalamus. Factors that promote growth hormone secretion include deep sleep, hypoglycemia, exercise, amino acids, and ghrelin. Factors that inhibit growth hormone secretion include elevated glucose level, IGF-1, and growth hormone. They inhibit growth hormone secretion by inhibition of growth hormone releasing hormone release and increased release of somatostatin. Please note, except deep sleep and IGF-1, most of these other factors that regulate growth hormone secretion are identical to those that regulate glucagon secretion. Factors that control growth hormone secretion are summarized in this slide. This chart shows you growth hormone levels are lower during the day but higher during sleep. There's an overall increase in growth hormone levels during puberty. Growth hormone release is stimulated by deep sleep. Deep sleep is also called non-rapid eye movement sleep.
Now, let's move to part 4. We will look at other factors for growth.
Androgens are anabolic hormones. Androgens act on muscle, increasing muscle size. Estrogen acts on the bone and maintain the bone. Estrogen and androgen also stimulate growth hormone release. The calcium level in the blood has a strong impact on bone homeostasis. Vitamin D and parathyroid hormones are the two hormones that regulate bone growth by control of calcium level. Thyroid hormones control synthesis of both growth hormone and growth hormone receptor. Fibroblast growth factors, or FGFs, are local regulators of growth and differentiation, especially in the bone.
Now, let's move to part 5. We will look at clinical correlation.
The first condition is pituitary dwarfism. It is caused by a deficiency in growth hormone production from the pituitary gland. The deficiency results in delayed growth and short stature, but the body development is proportional. Second condition is Laron syndrome. Patients with Laron syndrome have short stature and increased sensitivity to insulin. It is caused by a mutation in growth hormone receptor. The third condition is acromegaly. Patients with acromegaly have abnormally large hands and feet. They are abnormally tall, with pronounced brow protrusion and lower jaw protrusion. Acromegaly is caused by overproduction of growth hormone during adulthood after epiphyseal plate closes at puberty. This is different from gigantism, which is caused by increased level of growth hormone before epiphyseal plate closure. The fourth condition is Russell-Silver syndrome. Babies with this condition have a low birth weight and often fail to grow and gain weight at the expected rate. So this is a normal girl, and this is a girl with this condition. Head growth is normal. As a result, the head may appear unusually large compared to the rest of the body. Russell-Silver syndrome is caused by a loss of IGF-2 expression due to defects in genomic imprinting. Here is integration with genetics. IGF-2 gene is imprinted differently in paternal versus maternal chromosomes. Defects in printing leads to loss of IGF-2 expression. An opposite effect is seen in Beckwith-Wiedemann syndrome. The last condition is achondroplasia. Patients with this condition have short stature, prominent forehead, and low nasal root. It is caused by a point mutation in FGF receptor 3 gene. The mutation causes constitutively activation of FGF receptor 3, which inhibits bone growth.